and the first two topics, important considerations and critical factors for RNA preparation, are describing more general aspects of RNA preparation which should be considered. These aspects have been discussed more extensively in former webinars, and I will touch this not very deeply, will give more an overview. Here I have listed some challenges you will be faced when working with RNA, and these aspects need to be addressed when you work with RNA. These are the sample handling before extraction. Is it a critical sample type I'm working with, and which purification protocol must I use? What kind of quality control do I need, or what efforts do I have to run for it to get the right information? And the last aspect is to keep in mind, do I need, for instance, a long-term storage of my RNA for later experiments, or when I want to confirm these experiments in a later experiment? Here you can see the whole workflow, which is divided into pre-analytical steps, this on the left-hand side here, and into the analytical steps where the molecular analysis will be done. The quality control steps are more between. And here I have listed some issues or problems that could arise in the different steps, and I don't want to go into detail into these different issues which have been addressed in webinars before, but more into the quality control. And these questions that arise are: how to quantify my RNA? How can I assess any contamination, either gDNA or other contaminants? And how can I check for my RNA integrity? The question is, or the problems that could arise: wrong quantification could lead to over or underestimated yields, or if I don't assess the integrity, maybe I observe low integrity. Here you can see a variety of different biological starting materials and a related question how to collect and to stabilize this material in a proper way. As an example, I would like to choose whole blood or plasma as a sample type. And for the collection, you have to consider to use a blood collection tube with the right volume and that it is compatible with your sample preparation method. What does it mean? That means try to prevent, for instance, heparin, because this anticoagulant can interfere with downstream assays like RT-PCR, for instance, which means that a doctor in the hospital collecting your blood must be aware thereof and using the right collection tube for your purpose. Similar considerations have to be done when working, for instance, with cellular samples, and here I don't mean cultured cells. But for instance, epithelium cells collected with swabs or smears collected with brushes, for instance. As a consequence, choose the right collection device and stabilization method for your samples. These are all issues and questions or aspects that have to be considered in the pre-analytical phase of your experiment. In the next part, I will point out some critical factors for the RNA extraction step itself. The RNA yield can be impaired by, for instance, an inefficient lysis of the sample material, independent if the starting material are cells, tissues, or blood. Also keep in mind that the mRNA content is only 1 to 5% of the total RNA amount, and therefore choose the right purification method for the analyte of interest. For instance, if you want to purify total RNA with or without, for instance, the small nucleic acid fraction, like mainly miRNA. The integrity and purity of the RNA will be affected by the quality of the starting material, inactivation of Rnases, and I will give a brief overview how this can be achieved and what should be noted. Purification of potential inhibitors like heparin in RT-PCR applications. I mentioned this before. in general, protein, respectively nuclease contamination, and genomic DNA contaminations, and I will come back later to this point. The inactivation of RNases is a very important step, and normally RNases are denatured by using chaotropic salts like guanidine isothiocyanate or guanidine hydrochloride, which is part of the lysis buffer. In this case, RNA is normally, or the RNA in the homogenates, can be stable for several hours at room temperature. Sometimes there might be sample types that have higher content of RNases, and complete inactivation of RNase activity is achieved using a reduction agent like beta-mercaptoethanol, this is shown here, or D-thiothreatol, shortly DTT, it's shown here, to completely denature RNases, which is indicated in this reaction. So the combination of chaotropic salt and a reduction reagent leads to a denatured reduced ribonuclease. I would like to show you some experimental data addressing this issue. And first, I would like to show you an inhibition test of RNase A. This is shown here. We have mixed a lysis buffer with two different amounts of purified RNase A in the presence of mercaptoethanol and absence of mercaptoethanol, and made a repurification of spiked-in RNA. So the dark bars represent the samples plus mercaptoethanol, and the light bars represent the samples without mercaptoethanol. Afterwards, we analyzed the integrity of the RNA using an Agilent bioanalyzer and determined the RIN value for each sample. You can see that in the absence of mercaptoethanol, with increasing amounts of RNase A in the sample, the RIN value decreases. You can clearly see this from here to here. In the presence of mercaptoethanol, we obtained a good RIN value. This is shown here. Which is comparable to the control without the additional or the addition of RNase A. This is shown here. We made an additional experiment testing the efficiency of different reduction agents. And here you can see data for the purification of RNA from peripheral blood mononuclear cells, PBMCs. These cells will be isolated from whole blood, and this sample type is sensitive to RNases. We performed a standard easy RNA extraction procedure in the presence of different reduction reagents. This is shown here in the presence of mercaptoethanol or DTT. With different concentration, with decreasing concentrations. The readout was also the RIN value as an indicator for RNA integrity. There's nearly no difference for the extractions using either beta-mercaptoethanol or DTT at the recommended concentrations. In contrast, TSAP, a phosphine-based reduction agent, is not sufficient to prevent RNA degradation and not sufficient to inhibit RNases. The control without the addition of reduction reagent is degraded. You can see this here. The electropherograms indicates the integrity of the different sample, and for the control reaction, this is shown here, without any addition of reduction agent, you can see there's only degraded RNA, indicated by a RIN value of 2.5. With the addition of beta-mercaptoethanol or DTT at a recommended concentration, you obtain high-quality RNA from the sample type with RIN values of 9.9 and 10. The addition of TSAP is not sufficient to prevent degradation of the RNA, resulting in an RIN value of 4.6. In the next couple of slides, I would like to look deeper into different aspects of judging RNA quality and integrity. The purity of RNA preparations are usually judged by OD ratios, measuring absorbance at 260 nanometers and 280 nanometers, or the ratio 260 and the ratio to 230, absence of contaminants, absence of gDNA, and the stability of LUAs. The integrity of RNA preparations are usually judged by gel electrophoresis, or more or less common capillary gel electrophoresis, like the QIAxcel from QIAGEN or the Agilent bioanalyzer. And the comparison of the 3'/5' ratio. The next topic I would like to discuss is the determination of the purity of RNA. This is usually be done by calculating the ratios of the readings at 260 and 280 and 260 and 230 nanometers. And these ratios can provide an estimate of the purity of RNA, but please consider the following aspects. The A260 and A280 ratio is influenced considerably by the pH. The ratio is ideally around 1.8 to 2.1 at pH 7.5. You will get lower ratios with solutions at lower pH. For accurate values, we recommend measuring absorbance in 10 millimolar tris Cl, pH 7.5, and pure RNA has a ratio of 260 to 80 of 1.9 to 2.1 in the solution. The ratio of absorptions at 260 versus 260 to 80 is commonly used to assess DNA contamination of protein solutions, since proteins, in particular the aromatic amino acids, absorb light at 280 nanometer. The reverse, however, is not true. It takes a relatively large amount of protein contamination to significantly affect the 260 to 80 ratio in a nucleic acid solution. That means the 260 to 80 ratio has a high sensitivity for nucleic acid contamination in protein solution, but the 260 to 80 ratio lacks sensitivity for protein contamination in nucleic acids. For instance, 100% DNA has a ratio of approximately 1.8. And this difference is due to the much higher extinction coefficient that nucleic acids have at 260 and 280 nanometers compared to that of proteins. Because of this, even for relatively high concentrations of protein, the protein contributes relatively little to the 260 and 280 absorbance, while the protein contamination cannot be reliably assessed with a 260 to 80 ratio. This also means that it contributes little error to DNA quantitation estimation. Another important source for varying 260 to 80 ratios are phenol contaminations. The second criteria, which is quite often used to assess nucleic acid purity, is the 260 to 30 ratio. The expected 260 to 30 ratio for pure nucleic acid is often higher than the respective 260 to 80 ratio and is commonly in the range of 2.0 to 2.2. The 260 to 30 ratio can be affected by other common contaminants and as a consequence resulting in low ratios. Such contaminants are EDTA, carbohydrates, and phenol, which have absorbance close to 230 nanometers. Contamination by phenol, which is commonly used in nucleic acid purification, can significantly throw off quantification estimates. Phenol absorbs also at 270 nanometers and generates a A260 to 80 of 1.2. As discussed before, nucleic acid preparations uncontaminated by phenol should have a ratio of 260 to 80 of around 2. Therefore, contamination by phenol can significantly contribute to overestimation of DNA concentration. Absorption at 230 nanometer can also be caused by contamination by guanidine hydrochloride and guanidine thiocyanide. And guanidine thiocyanide absorbs at very high concentration, also at 260 nanometers. But what does it really mean? And I will give you an example. In this slide, I would like to show you the effect of guanidine salt contamination on the 260-230 ratio and what it means for real-time, for instance, for real-time PCR. Low 260-230 ratio is mostly due to cyanide carryover. Thiocyanide absorbs very strong around 220 to 230 nanometers. Guanidinium thiocyanide is present at very high concentrations in the lysis buffer or extraction reagent used in the most RNA purification procedures. Based on our experience, the 260-230 ratio of an RNA sample is strongly reduced when guanidine thiocyanide is present even at submillimole concentrations. Also, concentrations of guanidine thiocyanide of up to 100 millimolar in an RNA sample do not compromise the reliability of real-time RT-PCR even when using an inhibitor-sensitive PCR chemistry. And on the next slide, I would like to show you data addressing exact this issue. In this experiment, we have tested the effect of guanidine salt concentration on the 260-230 ratio and the downstream real-time RT-PCR application by adding the indicated amount of guanidine salts to the eluate and using an inhibitory-sensitive RT-PCR for beta-actin. The concentration of RNA in the eluate was always 50 nanogram per microliter. In figure A, this is this figure here, you can see a plot with the ratio of 260-230 for RNA samples containing either 0.03 to 100 micromolar guanidine hydrochloride. These are the purple dots here. Or for 0.01 to 30 millimolar guanidine thiocyanide. These are the blue diamonds. There's nearly no change in the 260-230 ratio for guanidine hydrochloride. You can see this here. Even with increasing amounts of guanidine salt, there's no change of the 260-230 range. The ratio is around 2. For guanidine isothiocyanate, there's a dramatic change in the 260-230 ratio and already at concentrations between 0.1 to 0.03 millimolar, it shifted to a ratio of 1. You can see this here. And to much lower ratios at higher guanidinium salt concentration. But how is the PCR affected due to this guanidine salt concentration in the eluate? This is shown in figure B. CT values were obtained from a real-time one-step RT-PCR using a TaqMan gene expression assay for beta-actin and a master mix from a different supplier, not QIAGEN. The reaction volume was 25 microliter and the template were either 1 microliter or 2 microliter of a 50 nanogram per microliter RNA sample containing 0.3 up to 1000 millimolar guanidinium salt. Inhibition of RT-PCR is observed at concentrations around 100 to 300 millimolar and higher. The correlating 260-to-30 ratio for guanidine isothiocyanate at this concentration would be below 0.5. That means with a 260-to-30 ratio around 1, this is still more than 1 order of magnitude before inhibition is observed. In the next slide, I would like to show you the effect of phenol on the absorbance at 260 nanometers. In the left panel, you can see how the absorbance at 260-270 nanometers of an RNA solution increases even at low amounts of phenol in the sample. This is the curve for pure RNA with an absorbance maximum at 260, and the same solution contaminated with phenol with an absorbance maximum at 270 nanometers. In the right panel, there is a wavelength scan only for phenolic solution, and there you can see the absorbance maxima at 230 and around 217. As a consequence, a phenol contamination will imitate higher RNA content of the sample. But what is the influence of remaining phenol in your RNA sample in a downstream application like PCR? This is shown in the next slide. In this experiment, I will show you that the purity of nucleic acid has a critical effect of sensitive downstream applications. Contaminations like phenol remaining after RNA purification can dramatically reduce the efficiency of your downstream application. In this experiment, total RNA was purified from rat muscle using the RNeasy fibrous tissue minikit, and in comparison, a phenol guanidine containing reagent. Quantitative real-time PCR was performed using the Quantitech probe RT-PCR CAT and primer probes for CTON, a transcription factor. CT values are shown with triplicates for each RNA amount. You can see that with increasing input template RNA amounts from the phenol-based extraction procedure, this is the blue curve, there's no linear correlation for the CT values starting with 10 nanograms and higher. The CT values keep the same despite higher RNA input amounts. In contrast, using the RNA extracted with the RNeasy fibrous tissue minikit, the assay shows a good linear correlation. This is shown by the red curve. Therefore, phenol remaining after RNA purification can dramatically reduce the efficiency of your downstream application. And for sensitive downstream application, it's critical that any contaminant is efficient and reliable removed during your RNA purification. Another challenge for RNA purification is the contamination with genomic DNA, and I will discuss this in the next slide. Here I have listed consequences of genomic DNA presence that could arise. Presence of genomic DNA in your purified RNA fraction can lead to overestimation of RNA yield, overestimation of transcript abundance in RT-PCR, especially if you look for raw or low abundant or low copy transcripts. And it can lead to higher background, for instance, in microarray or NGS experiments. Therefore, it's very important to get rid of gDNA contamination from your sample. And in the next slide, I will describe some approaches how you can achieve this. There are different ways to eliminate a genomic DNA during the sample preparation itself, but also afterwards. The first method is based on treatment with DNAs. This can be done either on membrane, if you use, for instance, a spin column-based method, or on bead, or in solution. This method is more efficient than on membrane, especially for large amounts of DNA, and a higher efficiency is mainly for sterical reasons. The second method is based on chemical separation of DNA and RNA. This can be achieved by organic extraction, using acid phenol chloroform like the QIAxcel reagent, or by specific binding of the genomic DNA to a solid matrix, like the gDNA eliminator spin column in the RNeasy Plus mini kit. The third method for genomic DNA removal can be a built-in step, for instance in the cDNA synthesis protocol, as we offer in the QuantiTech reverse transcription kit. However, combinations of the mentioned different methods are also possible. An example for the efficient gDNA depletion will be shown on the next slide. In this experiment, RNA was extracted from PBMCs. I mentioned this before for another experiment, so the same sample type, and compared to competitor kits for RNA yield and gDNA depletion using the RNeasy Plus mini kit. This kit includes an additional spin column for the chemical separation of genomic DNA. In the left figure, this is shown here, you can see that the RNeasy Plus mini kit provided the most consistent RNA yield with lower variances compared to both competitor kits, and also the higher yield. You can see this in this table here compared to the other competitors. The residual DNA amount was assessed by real-time PCR amplification of the HOXD9 gene, revealing an approximately 10-fold better elimination of gDNA contamination compared to the competitor kits for gDNA elimination. You can see this here. The RNeasy Plus kit has a much lower residual DNA content compared to the other competitors. The quality assessment was done using the QIAexpert Agilent bioanalyzer, resulting in comparable to 60:80 ratios. You can see this here in this table. Values around 2, as well as comparable RIN values indicating high quality RNA, and also the RIN values showing or indicating very high intact RNA. In the last section of the RNA quality control part, I would like to show you that poor high quality RNA can be stored for a prolonged time, so that you can perform experiments with the same RNA afterwards to a later time point. Here you can see the result of a storage test of RNA that was purified from cultured eukaryote cells. And the integrity of the extracted RNA was analyzed using an Agilent bioanalyzer and repeated after 18 months storage at -20 degrees of the RNA. Comparison of the 2 time points reveals that either the ratio of the ribosomal RNAs, 28 and 80S, as well as the win values are identical after the storage time. The same test was done using tissue samples and shown on the next slide. The same experiment using rat spleen tissue was performed, and you can also see even with tissue sample there is no significant change in RNA integrity observed after storage, after 18 months storage at minus 20 degrees. We also obtain high quality RNA. To summarize, the important aspects with respect to the storage of RNA are: pure RNA can be stored at minus 80 or minus 20 for prolonged periods. However, try to avoid repeated freeze-thaw cycles. If possible, store the RNA fraction in aliquots and use low binding tubes. Under this condition, no degradation of RNA is detectable even after 18 months. In the next section, I would like to give you a general overview about the importance of RNA integrity. The term integrity refers to how intact and undegraded your RNA is. A high integrity is important to obtain an accurate and quantitative measurement of gene expression at the moment of RNA extraction. As I mentioned before, RNA integrity is limited mainly by the quality of the starting material, but also, of course, by the right storage and stabilization conditions. The 3'-5' ratio may represent RNA quality. With this ratio, you compare signals obtained from amplicons, for instance, generated with RT-PCR or with capture probes using a microarray at different distances from the 3' end. For instance, here, here, and here. A PCR reaction of intact RNA will result in roughly equal signals for all amplicons for the target mRNA. Whereas partially degraded RNA, the cDNA synthesis will generate shorter or less fragments in proportion to the degree of degradation. So for instance, depending on the degree of degradation, you will obtain longer or shorter amplicons. On the next slide, I will describe some tips how to check the RNA integrity. Check the intensity of the ribosomal RNA bands on either an agarose gel or the more common method in the meantime, capillary electrophoresis. I will come later in this section to the capillary electrophoresis-based methods. In RNA from eukaryotic cells, the 28S band should be double the intensity of the 18S band. This would be ideally 2 to 1, but it's difficult to determine. In bacterial cells, check the 23S in relation to the 16S RNA bands. If the ribosomal bands or peaks of a specific sample are not sharp, but appear as a smear towards smaller sized RNA, that means it's likely that the sample suffered major degradation either before or during your RNA purification. However, agarose gels are still be used for the check of RNA integrity, but capillary electrophoresis-based methods are more common, and I will describe these methods in the following section. In this section, I would like to present the actual state-of-the-art methods for the quantification of RNA and for the assessment of RNA integrity. In the sections before, I discussed different aspects and criteria that are important for RNA quality control. These criteria are listed and summarized in the table below in detail. Determination of protein contaminants using the 260-80 ratio. Salts and other contaminants using the 260-30 ratio. Yield, sample integrity, size range, quantity of double-stranded DNA versus other nucleic acids. And these are instruments or methods capable to assess the different QC criteria. For instance, the nanodrop, agarose gels, the qubit fluorimeter, the QIAxcel Advanced instrument, and the QIAxpert spectrometer. No single instrument, as you can see, can cover all QC parameters, but a combination of the QIAxcel Advanced and the QIAxpert covers all RNA QC parameters. Actually, there is no one-for-all solution in the field available. In this experiment, we compared the measurement accuracy for 3 different methods. For this, we used a commercially available reference total RNA at a certain concentration, and a total of 40 replicates were measured each on the QIAxpert I mentioned before, on a nanodrop, and a qubit system. You can see that the QIAxpert delivers the lowest coefficient of variation value for the measurement of the 40 replicates. The highest variation was obtained with the qubit, and a lower variation was also observed with the nanodrop spectrometer. The same RNA sample was used to prepare a serial dilution and to compare the linearity of the measurement compared to the nanodrop, shown here, and the qubit. The QIAxpert shows an excellent linearity between the expected measurements or the expected values and the real measurement. For the nanodrop and for the qubit system, you obtain a systematic overquantification from the sample compared to the sample value should have been. In the next couple of slides, I would like to present the RNA integrity score to assess RNA integrity using the QIAxcel Advanced System that enables fully automated sensitive high-resolution capillary electrophoresis. The total RNA quality is assessed on the QIAxcel by analyzing the migration pattern of your sample and the 28-18S ratio to interpret the integrity. And I guess some of you know this picture or a similar picture from the Agilent bioanalyzer. The advent of capillary electrophoresis provides the basis to assess RNA integrity based on the migration pattern. The RIS and the RIN values are based on a large collection of electrophoretic RNA measurements and used to extract an algorithm that describes RNA integrity. The RNA integrity score has been developed from QIAGEN using the QIAxcel, and the alternative RIN value is the RNA integrity number from Agilent's bioanalyzer. Both are indicators reflecting RNA integrity and are intended to predict the validity of downstream qPCR. Both the RIS and the RIN values range from 1 to 10, where 1 represents highly degraded RNA and 10 represents highly intact RNA. The system analyzed several different electropherogram parameters, as I mentioned before, including the 28S and 18S peaks analysis and a migration pattern. Usually values between 7 and 10 are indicators of RNA quality suitable for downstream applications. Of course, this depends on what is achievable with the sample, and for instance, RNA from an FFPE sample, it might be difficult to achieve that risk values. You will obtain lower. The risk value allows comparison of sample, standardization, and repeatability of experiments. And in this slide, 3 samples of different quality are shown, and the RNA was extracted from rat liver tissue, and the sample A1 on this line, the red curves, is highly intact and has a risk value of 9.5. Sample A7, the blue one, you can see this in the chair-like image here, is partially degraded and has a risk value of 5.8. Sample 11, the green sample, is strongly degraded. You can see this here towards the smaller fragments, and has a risk value of 3.6. The respective electropherograms are shown on the right, and in the superimposed view you can see how the sharp peaks from the 28 and 18S signal decreases, and you obtain a broad region in the green for the green sample, indicating highly degraded RNA. In this slide you can see a direct comparison between the QIAxcel and Agilent bioanalyzer, and total RNA. Oops. Sorry for that. And total RNA from Jurkat cells were extracted, and decreasing integrity levels. The integrity values range from intact to degraded, were analyzed on the QIAxcel Advanced, you can see this here, the gel-like image, and on the Agilent bioanalyzer. The RIS and the RIN value numbers for the samples are listed here in the table, and you can see there's a good correlation between the values for the same samples, for either the QIAxcel or the Agilent, but also for the duplicates. To demonstrate you that this is a very good correlation between both systems in general, we made a correlation of QIAxcel RIS and Agilent RIN values from a large number, more than 350, of different sample types, like different cell cultures or different tissues like liver, kidney or so. And this is shown here in a dot plot below. And the coefficient of determination, R square, indicates the degree of correlation. As closer it gets to 100%, the better is the correlation between both data sets. For this kind of statistics, an R square of around 93% is very high, meaning we have a very good correlation between both systems. This is another example I would like to present. Total RNA was extracted from rat liver samples using the RNeasy Plus mini kit and subjected to different decomposing conditions, meaning the RNA was degraded in an artificial manner. Samples of increasing levels of degradation, you can see this from here, from the left to the right, were analyzed on the QIAxcel Advanced and the Agilent bioanalyzer. And the obtained RIS and Rn numbers are listed in a table here on the right-hand side. And you can see that we also, for this example, we obtain a good correlation. This means the RIS provides objective quality measurements for RNA samples. It eliminates the need for human interpretation and enables implementation of rigorous quality control and gives highly reproducible results comparable to the Agilent bioanalyzer. And we are at the end on our webinar. I hope I could give you an overview of the different aspects with respect to RNA quality control and how important it is to use the right quality control parameter depending on your scientific questions. Furthermore, our results demonstrated that the QIAxpert and QIAxcel covers all important quality control parameters and that there is a good correlation with other methods like the Agilent bioanalyzer. I would like to thank you for your attention, and yeah, I'm happy to answer your questions.
Yeah, thank you so much, Peter. I think that was very informative for me as well, sitting here. I can see some questions dropping in. So the first question that I have on the table is, so you talked about the 6 parameters for the RNA quality control. Mm-hmm. The question is it important to check all of them, or can you live with 1 or 2?
This is a good question, of course. Well, for instance, if you have an extraction procedure where you use a phenol containing reagent, you should do the 260-230 ratio. Just to be sure that there is no overestimation or interference to expected with your downstream application. If you have a standard RNA extraction, let me say from cultured cells, and you know you're using a guanidinium hydrochloride based extraction reagent, you will not obtain an influence on your 260-230 as I showed. There is no, even at high concentrations, there will be no influence. So it depends on your setup.
Okay, great, thank you. The other question is, can DNase treatment be enough to assure the genomic DNA removal at the isolation step, or would you recommend to use the combinations as you showed on your slides?
I would say a clear yes to that DNase treatment on column is sufficient. It depends on the expected yield. So if you know you will end up with a very high RNA yield, it might be better to include a second method. But if you obtain not so much, the on-column DNase digestion is fine. So using the right DNase, you will end up with a nearly DNA-free sample.
Okay, great. The another question, this is a very specific one, I would say. The person wants to know what is the best sample anticoagulant. Is there a specific type for if I understand the question, Eppendorf's for storage? Yeah.
Yeah. Okay. I think I got the question. Just a short additional information. If you want to work with blood or want to prepare plasma from your blood and you rely on a doctor in a hospital, it's very often that these doctors have a fixed set, which means they are using a fixed set of blood collection tubes. And in a bad case, they're using heparin because any downstream assay for a clinical parameter needs heparin treated blood, and then you have to live with this heparin. So it's very important to check up before you start with your experiment what is the source of your collection blood. And for that reason, there should be no problem with EDTA or citrate-based collection tubes for molecular analysis.
Okay, great. I think we're running a little bit short on time, so I just have time to take one more question. For the others, because some questions are that will you get the slides? Yes, we'll try to upload them, and you can also request them with us. We will try to upload the recording of the webinar, but if you miss something, we will repeat this webinar series in October as well, along with the other sample preparation and the integrity part from FFP samples. So the last question that I can take today, and the rest we will try to answer you back via email. Either myself or Dr. Peter we can definitely get back to you. So apologies for that. So last question is, will you please explain the RNA degradation and how it can be recognized that at 28S or 18S level, because this is still confusing for this person. I don't know if yeah.
Yeah, okay. Well, with artificial RNA degradation, I mean your RNA can be degraded by different methods or mechanisms. For instance, heat-induced degradation, or RNase-induced degradation, or for instance light or UV induced degradation. And normally if you have an agarose gel, you can see that the 28S band has a higher intensity of the 18S band. This is always a good indicator using an agarose gel. If you see that the 28S band is less intensive than the 18S band, and you have a more intensive smear or migration pattern below the 18S band, then you have an indication for degradation. And if you perform a PCR, an RT-PCR assay, you should be aware of how to synthesize the cDNA and at which position of your target gene your assay is located. Then a 3' or an assay close to the 3' assay might be better than to the 5' end.
Okay, great. I think that's it from our side, and thank you again everyone for joining today's webinar. We look forward to having you for the next in the series on next Monday, and I wish everyone a great day, and thank you again, Dr. Peter Porshwesky.
Yeah, a pleasure, my pleasure. Bye.